Muhammad Umar Farooq Biological Video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also question to each other for solve the problem. So today we will discuss about the basic light reaction to dark reaction in photosynthesis. Then sugar to starch synthesis in amyloplast. So first of all, the chloroplast is used for making sugar, and that's go into the amyloplast to synthesis. So basically in cytoplasm is also, but here we will discuss, here is the cell which that is contain chloroplast and mitochondria as well as nucleus. So there are many organelles are present in the cell. So here is the cell wall, and there's chloroplast and cytoplasm which that is present inside of the cell. So the bigger vacuole is also present. So here is the smaller, but that will be bigger. So here's the mitochondria. Now here I am drawing the chloroplast, which that is contain a double membrane organelle, that is double membrane organelle, which that's contain a thylakoid with a combination of granum and is known as grana, and that's outer and inner cell, outer and inner chloroplast membrane, which that is surrounded by the stroma inside, that's floating the thylakoid into the stroma. So here I am drawing the thylakoid membranes and thylakoid granum. So three granum is present, is known as grana. So here is the membrane of the granum contain chlorophyll, and this chlorophyll contain also light absorption, and there are many complexes which are used for the production of NADPH, as well as ATP synthase enzyme used to produce ATP, and NADP plus reductase used to NADPH production. So there are many RU reaction. So here we are recapping. So here RUBP enzyme most abundant in the world, and Rubisco enzyme are present into the stroma, which that is used for the fixation of carbon dioxide and convert it into glyceraldehyde 3-phosphate, which that is the precursor of sugar. So here we will discuss about the stroma, which that is used for the Calvin cycle. That's the Calvin cycle is the dark reaction, is not light dependent reaction, but it's depend passively because NADPH and ATP is used for it. So here I am drawing the ribulose 1,5-bisphosphate. This is also known as RUBP, or Rubisco enzyme. This enzyme is fix the carbon dioxide in the phase of carbon fixation phase, and this carbon fixation phase, in this way the 3-ribulose 1,5-bisphosphate convert into 6,3-phosphoglyceraldehyde, and that 6-phosphoglyceraldehyde is 3-phosphoglyceraldehyde because carbon number 3 contain phosphate group, and these 3-PGA is central metabolic pathway for use it. So remember about that. Here is the carbon fixation, and now we will discuss about the phase 2, which that is the reduction. And in this phase the one ATP is used, 6 ATP used to produce a 6 ADP, and that's the product will be 1,3-bisphosphate glycerate, and that is the 6 molecule 1,3, carbon number 1 and carbon number 3 contain a phosphate group. And here we will discuss is here that is the NADPH is used for the NADPH for the dehydrogenase enzyme to convert NADP plus, and that the product will be glyceraldehyde 3-phosphate. It means the G3P, and this G3P is 6 molecule, one molecule will be separate one by one, and again cycle will be continue, and again carbon dioxide fixation. And these phosphate, inorganic phosphate group, release from the dehydrogenase enzyme, and it is known as reduction, because reduction means reduce the glyceraldehyde 3 phosphate. So here I am drawing the one glyceraldehyde 3 phosphate will be separate, and other 5 molecule will begin to produce by the using of ATP, 3 ATP into 3 ADP to convert 3 ribulose 1,5 bisphosphate, again a Ribisco enzyme. So this is known as regeneration, because regenerate again Ribisco enzyme for further process, for further further reducing of the glyceraldehyde 3 phosphate. So the major thing is the during reduction phase, that is the molecule of glucose, one glyceraldehyde 3 phosphate, which that will be separate, and this is the precursor of making synthesis of glucose. So now we will discuss about step by step after the Calvin cycle become done, and separation of the glyceraldehyde 3 phosphate. So one molecule of sugar will be responsible for the making of basically two molecule of glyceraldehyde 3 phosphate is used to make one molecule of glucose, that is 6 carbon. So here is the 3 carbon containing precursor molecule of product, which that is glyceraldehyde 3 phosphate. These molecule will be different production by the synthesis. So here is the protein, which that is present in the beans in plant, which that is produced by the proteoplast, and the olive, sunflower, castor bean, which that is store the fat by the basically lipoplast, which that store these. And on the other hand, the grains are wheat, which that is store starch into the amyloplast. So amylose and amylopectin is the major source of the starch. So here we will discuss just starch, and that is the amyloplast into the amyloplast. So another rest of the two reaction we'll discuss in the next video. So here is the chloroplast, which that is produce glyceraldehyde 3 phosphate by the Calvin cycle. So this Calvin cycle will be produce glyceraldehyde 3 phosphate by the one cycle. So the one cycle will be done, and again one cycle is more to produce 2 glyceraldehyde 3 phosphate or G3P, and these 3 carbon containing molecule will attach to each other by aldolase enzyme. So 2 molecule will combine to produce fructose 1,6 bisphosphate. So these fructose 1,6 bisphosphate mean bisphosphate mean one carbon number 1 and carbon number 6 contain phosphate group in this fructose. So this fructose is further processed, and the major enzyme used for it, fructose 1,6 bisphosphatase. So these phosphatase enzyme used to release the inorganic phosphate, and this phosphatase enzyme is used for the inorganic phosphate releasing. So these inorganic phosphate will be released and produce again fructose 6 phosphate. So these fructose 6 phosphate I will write here. So remember about these things. So the fructose 1,6 bisphosphate phosphatase enzyme to produce by the releasing of the inorganic phosphate to convert fructose 6 phosphate. So these fructose 6 phosphate is convert by the reversible reaction, which can be reversed, but it's here is the reversible reaction for the further production of some sucrose. So here we can say that the fructose 6-phosphate can also be converted into the sucrose, which that are present in the sugarcane. So remember about that things if reversible. So here is not reversible, is going forward, and phosphoglucose isomerase enzyme used to produce glucose 6-phosphate. So these glucose 6-phosphate phosphoglucose isomerase to produce uh, glucose 1-phosphate. It means the carbon number 1 contain again the phosphate group. In this reaction, phosphoglucomutase enzyme used for the mutation of the molecule. Mutation means the changing. So the location will be changed from the carbon number 6 to carbon number 1. So here is the glucose which that is bind by the So here is the glucose 1-phosphate which that is carbon number 1 contain phosphate group. So the major important reaction will begin here. So the AMP as adenosine monophosphate will become toward the glucose 1-phosphate, and these adenosine monophosphate which that's contain adenosine and fructose which that's come together and attached by the glucose 1-phosphate. So attaching of these is looking like here. So this molecule glucose glucose 1-phosphate which that attached by the here is known as adenosine diphosphate glucose. So the glucose will release glucose will be detached from it and give the phosphate group toward the AMP to convert ADP. So this ADP is the adenosine diphosphate. So the changing of these, that's a giving of the phosphate group toward the ADP from the AMP convert into the ADP, and that is ADP is again AMP is used for further this reaction from the previous again, and that will be continuous, and that is the amyloplast is used for it. So that's amyloplast which that is use this sugar, and this is the important things which that is for example here I will draw another glucose molecule which that is come from the one more reaction of this glucose production. So here I will draw one more glucose molecule. Then we will see about the combination of these two glucose molecule to convert further more and more glucose molecule combination and their condensation reaction which that is used for the releasing of the water, and that is the glycosidic bond is produced. So here is the two molecule which that is attached to each other. How it's possible? It's possible due to the starch synthase enzyme. This starch synthase enzyme is used to release the water from here, and glycosidic bond will be produced. That's glycosidic bond is important, the glucose to glucose molecule combination bonding, and that's oxygen is the between the bonding. So here is you can see that and you can watch this diagram to understand that will. And this is the reversible reaction, because if water will be add again and that's convert into the simple sugar, and their reaction is similar. And more if we will talk about the fructose, if fructose is detached from the fructose reaction will be continue, and that's fructose to glucose combination to produce sucrose in the sugar cane, and that's the important things. And here is you can say that the formula of the formula of the basically glycosidic bond is here, that is the formula of the amylose. And amylose is the chain of the single chain of the glucose which that is showing in the figure. If we will talk about the branches of this chain, which that is arise the branches from the single chain to multiple chains or multiple branches, and these branches is convert into the amylopectin. So remember about that things. Here is the single chain I am drawing again, and that single chain is known as the amylose, and these again conversion into the simple sugar by the using of enzyme is known as amylase, and that's amylase enzyme convert into the dipeptide and oligopeptide oligosaccharide and oligopeptide. So oligosaccharide and disaccharide, which that is further convert into the disaccharase enzyme and oligosaccharase enzyme. So here is the amylopectin, which that is in the form of branches, and these branches is basically is due to the more further starch synthase enzyme used to it to make it branches. So these branches are present in the form of fiber like structure. Basically here I will draw a structure of starch, which that's also the amylopectin and amylose formation. So the amylopectin and amylose is the single same. So yeah, here I will draw a different form of amylopectin and amylose product, which that is produced by this reaction, and this reaction is some processed food, which that is also the part of these reaction, this result, because here is the different type of the uh, product. Here is the rice and bread, wheat, burger, and here is the zea maize and triticum aestivum, and here is the beans and bread and burger is the processed food, while rice, beans, wheat and corn, or you can say that the scientific name is zea maize. And if we will talk about the wheat, which that is the um, uh, basically triticum aestivum, and if we will talk about the rice, which that is the name of scientific name is Oryza sativum. So this all thing is basically is product from the starch synthase enzyme used to produce it. So that's all reaction is. I hope you make understand about this video, and if you like this video, please subscribe my channel and like my video and share to your fellows. Thanks. Bye.